Hi everyone, thanks for checking out this video. Okay, so now that we've reviewed a little bit of genetics, it's time to start getting into the actual material for the second project. So the second project in this course is going to be doing something called a genome-wide association study. So you guys might have heard of this before. It's commonly called GWAS for short. And yeah, I'd say it's one of the most common, like most famous types of analyses in bioinformatics. And yeah, it comes up a lot. It's a very common type of study. And yeah, so the second project is going to be doing this type of analysis on a mock like fake data set. And so the reason we're using a fake data set for project 2 is that like basically in project 1 was about gene expression data, and gene expression data, like I showed you guys, is pretty easy to find like publicly available. Like you can download data sets that are like real gene expression data sets on like government websites. Basically they just have like repositories on there. But for project 2, this type of data we're going to be working with, like genetic or genomic data. It basically has to do with like people's sequences of DNA, basically. So it's harder to find publicly available because there's like privacy concerns with it. People understandably don't want their like DNA sequence data just available to anyone who wants to download it on some like government repository website or something. So basically what I did for this project was I made up a fake data set, just like a mock simulated data set, but I made it somewhat realistic in the sense that the fake data set will be giving us the same type of information that we'd be working with if we were doing this type of analysis on like a real actual data set. So basically the fake data set is a little bit similar to the real thing in terms of what information we'll have access to and what type of analysis we'll be doing on it, basically. So with that being said, yeah, the topic for project 2 is going to be this field that I made up called zombology. So zombology, like you guys can probably guess, is the study of zombies. And yeah, we're going to be analyzing like a mock genomic data set related to zombology. So I have this like scenario for you guys to think about. So let's say that it's like a post-apocalyptic world where you're in the world after a zombie apocalypse has occurred, and you're in this like post-zombie apocalypse world, and you notice that when some people are bitten by a zombie, they turn into a zombie themselves. But then other people, when they're bitten by a zombie, they don't turn into a zombie. They just stay normal. So we're going to call the people who turn into a zombie after getting bitten the zombie phenotype. And we're going to call people who get bit and then don't turn into a zombie the normal phenotype. And the goal is going to be to analyze genomic data to look for SNPs, which, if you guys remember, the SNPs are just the one-letter differences in DNA. We're going to look for SNPs related to the zombie phenotype. So this is basically like the setup of a GWAS study in general. Basically, you have some trait that you're interested in, and the goal is to do like a scan of the genome, looking at these different SNPs, which again are just one-letter differences in the DNA. And yeah, and basically look for SNPs that are related to the trait that you're interested in. So for us, the trait, the phenotype, is whether someone turns into a zombie when they get bit by a zombie, or whether they just like stay normal after getting bit by a zombie. And yeah, so just as like another note too, movie recommendation for you guys. If you guys haven't seen this movie, by the way, yeah, I recommend checking it out because this is a awesome movie. But yeah, just put this here to showcase the zombie phenotype that we're interested in here. Yeah. So yeah, just again to review SNPs, because yeah, like in a GWAS, the SNP is really the object of analysis. So yeah, the goal is to like check a bunch of SNPs and look for the SNPs that are related to the trait in question. And yeah, so just to remind you guys again, this is a slide from the previous video. But yeah, when we're talking about alleles now, we're talking about alleles as being like different letters that could be at a specific coordinate in the DNA. So if you guys remember, yeah, we as humans have like 2 copies of every chromosome. So basically every chromosome and every gene within a given chromosome, we have 2 copies. So yeah, for every SNP, we have like two copies of that chromosome position. Basically, like we could talk about that position on each of our 2 copies of that chromosome, and then ask for each one, what letter do we have in that position? So maybe we have 2 A's, or we have 2 C's, or we have an A on one chromosome and a C on the other chromosome. Yeah, and again, I'm not, when I'm saying 2 copies, I don't mean 2 strands, because of course each chromosome has 2 strands where one strand is complementary to the other strand, but I mean 2 actual whole chromosome copies where each copy has the complementary strands. So yeah, like this whole chromosome here with its 2 complementary strands is one of the 2 chromosomes. And then the other chromosome over here is the other of the 2 chromosomes. And then yeah, talking about a SNP is basically talking about a coordinate in the DNA where there could be like more than one possible letter there. So yeah, if you guys see, most of these letters are just the same, but calling something a SNP means that there's a possibility of having different letters. Like maybe this person, like I said, has 2 A's, like an A on each one, or maybe they have a C on each of their 2 copies, or maybe they have an A on one copy and a C on the other copy, like in this picture here. And maybe it's also different combinations for different people in the population. That's basically the idea, is to basically, yeah, look at these SNPs in a population of people and try to look for SNPs where like a difference in letters in that position is associated with a difference in the trait that we're interested in, which in this case is whether someone becomes a zombie or not after being bit by a zombie, basically. Okay, so here's a little bit of information about our like fake mock data set we're going to be using. And in the next video, I'll show you guys how to actually like download it from my GitHub, and we'll start actually looking at the data set. But yeah, before we do that, this slide is just a little bit of information. So we're going to be analyzing a data set with 48,752 SNPs. And actually, so this is actually a pretty small data set compared to like the real data sets. Yes, this might seem like a lot, but it's actually pretty small as far as like the real data sets go. And the data set is going to have 100 normal samples. So basically 100 like normal people who didn't become a zombie, and then 100 zombies. So we're imagining in this scenario that we basically like sequence the DNA of yeah, like 100 normal people who got bit and didn't become a zombie, and then 100 people who got bit and became zombies, basically. And yeah, so it's fake data, but it's similar information to what we'd get in a real data set. And actually, I will tell you guys, I did simplify the format a bit. Yeah, the format is a little bit simplified compared to the format of these data sets usually in real life. If you guys want to see what the format looks like in real life, I'll put a link in the description to a video about like what these like file types, like how they actually look in real life. But yeah, even though it's a little bit simplified in terms of the format, to make it easy for us, like it does have the key information that we'd be interested in if this was like a real project, basically. Okay, so now for the hard part, and what I'm going to tell you guys next is going to be basically the hardest conceptual part of this second project. After we get past this part, things should be pretty easy from here, I think. But yeah, this is gonna be the conceptually the hardest part. So it might take a little bit of wrapping your brain around it. And if anything is confusing, by the way, just let me know in the comments and I'll try to answer. But yeah, the hard part is going to be basically understanding the format of this data and what this information is telling us, basically. And yeah, like what information we have to work with. This is going to be the hard part. And then once you guys understand what this table is and what all these numbers mean, basically. Once you guys understand that, yeah, it should be pretty easy going forward. But yeah, okay, so let's try to talk about what is being shown in this table and what information we have access to. Okay, so first things first, every row in this table is a different SNP. So basically every row is telling us in the first 3 columns some information about this SNP that we're interested in. And in the real thing, there's going to be like 48,000 or so rows of data where each one is one of the SNPs, basically. Okay, so let's talk about what these first 3 columns mean for each one of these SNPs. The first column is the SNP ID, which basically tells us in this like ID, like sample ID encoding, it basically tells us the chromosome number. So these are all chromosome number 1. If you guys remember, there's 23 chromosomes total, where we have 2 of each chromosome. So there's 23 chromosomes. 22 are like numbered chromosomes, like 1 to 22. And then there's the sex chromosomes, the X and Y chromosomes. So yeah, 23 different chromosomes, and we have 2 of each chromosome. So yeah, this is telling us, first of all, the chromosome number, where these first couple are all from chromosome 1. And then there's a colon, and then this number after that is telling us the coordinate of the position that we're talking about. So if you guys remember, just there's this sequence of DNA, and if you just imagine like starting counting at one end on like a reference chromosome, this second letter, this second number here tells us the coordinate of the position of this SNP that we're interested in. Basically, what position in the sequence of DNA on chromosome 1 this letter would be found at, basically. So yeah, this whole ID is basically telling us the chromosome number and then the like base pair coordinate position of the SNP, basically. Okay, on to the next 2 columns now. So these columns with the letters tell us the 2 possible alleles that could be at this SNP position, basically. So we have what's called our reference letter or our reference allele. Remember, we're using the word allele to just mean literally like one of the letters at this like SNP position. Like an allele here just means a letter in that position, basically. So we have our reference allele or our reference letter, and then we have our possible alternate allele or alternate letter. So yeah, just to further explain this, when we do DNA like sequence analysis, for each person that we like get the sequence of, we try to make sense of their DNA sequence as it relates to a reference genome, or like a reference sequence, which basically is just like a commonly used human DNA sequence. Actually, I think the real thing is like a composite sequence made of like not literally just one human, but a composite of like many different people. But yeah, we basically have our reference genome, and that's also where these coordinates come from, because the actual counts in the coordinate position could be like slightly different from person to person, but we get this like universal coordinate basically by aligning these people's DNA sequences to a reference genome, basically. So yeah, the reference genome tells us like the coordinate, and then it also tells us like for the reference genome, what letter does the reference genome have there basically. So in this case, for this first SNP, the reference allele is a T. So that's like the the reference letter in that position basically. And then the alternate allele is a C. So two possible alleles that could be in this position where the reference is a T and the alternate is a C. Where, again, just to stress this, the word allele here literally just means what letter is in that position. And yeah, we have our reference letter, which is a T, and the alternate letter, which is a C. Now, one like simplifying assumption I made when I was making up this like mock data set was I assumed, just to make things easy on us for this like practice project, I assumed that for every SNP, there was only one possible alternate allele. So yeah, I assumed that in this like SNP position, like the reference was a T, and I assumed that the only possible alternate allele was a C when I was making up this fake data. That's actually not always the case. You could have a SNP position, like a SNP coordinate, where maybe the reference is a T, and then one possible alternate allele is a C, and then maybe another possible alternate allele is an A in that position, or a G. So yeah, you could actually have in one given SNP position, like multiple possible alternate alleles. So that would be like a complication, because if that were the case, then the SNP IDs would not necessarily be like unique identifiers. There could be, if that was the case, like more than one like row in this table with the same SNP ID and like different possible alternate alleles. But just for the sake of like simplifying things for our project, yeah, I just assumed that for our purposes, every SNP only has one possible alternate allele, just to like make things easy on us basically. So whenever you see in my like mock data set, whenever you see yeah, like a SNP ID, we can treat these SNP IDs as unique identifiers, because I just assumed that every SNP ID only has one possible alternate allele. So in this position, you either have like the reference or the alternate on each one of your copies of the chromosome. Okay, so now for the little bit of the like maybe difficult, well, hopefully not, but maybe a little bit confusing part. What do these numbers mean here? Yes, I guess first I'll say too, like each one of these, like after the first three columns, each one of these following columns is one of the samples or one of the people that we like sequence the DNA of basically. And we have in their sample ID whether they were normal or a zombie. And we have 100 total normal samples in the real thing and 100 total zombie samples. But yeah, what are these zeros, ones, and twos? This is possibly kind of the confusing part. But yeah, I'll try to explain that now. We said before that, like, just like in the Mendelian model where each plant had like two allele copies, we also have like two alleles, like two allele copies, I should say, for each one of our SNPs that we're looking at. And by we, I mean humans. For every position in the DNA, we can talk about two copies of that position, because we have again two of each chromosome, so meaning like two of each gene, and also two of each like coordinate basically. So for chromosome 1 and then this coordinate, for this coordinate on chromosome 1, we have two copies of this of this coordinate position, basically. So yeah, we have 2 copies there. So for each person, it could be that both of their copies have a T, or it could be that both of their copies have a C, the alternate allele. Or it could be that one of their copies has a T and the other copy has a C. So are you guys following that so far? Hopefully that makes sense. Like we have 2 of each chromosome, so we have 2 of each gene. I should say too, it's not actually a guarantee that when we're looking at these SNPs, it's not a guarantee that all of these even are genes. These could be positions in the DNA that are between the genes, or that are just like unknown regions. So this is just like, this comes from the DNA sequence. These could be parts of the DNA that are within like known genes, or it could be within the unknown regions between the genes. Yeah, just positions in the DNA in general, including the space between genes. But yeah, we have basically, because we have 2 of every chromosome, we have 2 of each one of these chromosome positions, and then it's two possible like copies, where each copy could be each one of these letters in that position. So maybe both of our copies of this chromosome have a T, the reference allele, or maybe both have a C, the alternate allele, or maybe one copy has a T and one copy has a C. And these numbers here in this table, the 0s, 1s, and 2s, basically just tell us for that person, meaning that sample, like for that sample, did that person have, or sorry, basically it just tells us like how many copies of the alternate allele that person had. So a 0 means that person had 0 copies of the alternate allele for that SNP position. And by default, that then means they had 2 copies of the T allele, basically. Because you guys understand, because I made the simplifying assumption that there's only one possible alternate allele. Yeah, if they have 0 copies of the alternate allele, that means both of their 2 copies must be the reference allele, which is a T in this case. So a 0 here means 2 copies of the reference allele, because they have 0 copies of the alternate allele. If a person has a 1 in this table, that means that person had 1 copy of the alternate allele for that SNP. So this person had 1 copy of the C allele for this SNP position, meaning their other copy must have been a T. So if we see a 1 here, that means this person had 1 C and 1 T in that position. So yeah, one of their chromosomes had a C and the other chromosome had a T in that position. A 2 means the person had 2 copies of the alternate allele in that position. So this person having a 2 here means they had 2 Cs. So both of their chromosomes in this SNP position had a C. And yeah, we can also figure out from that they had 0 A copies. So yeah, 2 means 2 copies of the alternate allele, which in this case is a C. So we know that this person in this SNP position, both of their chromosomes in this SNP position had a C, and 0 copies for them were the A allele, basically. So yeah, I mean, maybe it's not so confusing. I don't know. You guys can let me know if you're following that or not. I hope it's not too confusing, but if that is confusing, just let me know in the comments and I'll try to further explain it. But yeah, basically just the thing to think about is for each one of these chromosome positions, we have 2 chromosomes. So we have 2 copies, or like, yeah, two allele copies for each one of these positions. And then we know what the reference allele is, meaning the allele on the reference genome that we're comparing it to. And we also know what the possible alternate allele is, where I made the simplifying assumption that there's only one possible alternate allele. So we know that, yeah, the reference allele and then the alternate allele. And then each person, each sample has 2 copies. So they could have 2 copies of the alternate allele, which would imply 0 copies of the reference allele, or they could have 1 copy of the alternate allele, which implies that their other copy is the reference allele, or they could have 0 copies of the alternate allele, which implies that both of their copies are the reference allele. And so basically, this data set is going to tell us the genotypes, where in this case, genotype just means what allele copies each person had for each SNP. And we're going to try to use those genotypes to see which genotypes or which SNPs are related to the phenotype of whether the person became a zombie or not after getting bit by a zombie. So basically, we're going to go through and like scan all of these SNPs and look for ones where the genotypes, these 0s, 1s, and 2s, are associated with the phenotype of whether the person was normal or a zombie, basically. So yeah, the next video we'll get started trying to do this analysis. Well, really, I should say the next video we'll focus on downloading the data set and like looking at the real data set before getting started. But yeah, more on that in the next video. So yeah, thanks for watching, and see you guys next time.